So welcome everybody to this session. If you've got the right session, it should be cutting edge analytics for the future. So the emphasis is on the future. We're thinking ahead and about the things that we need AI to do for us in data intensive healthcare. And we've got 50 minutes to talk about this. I'm Dave Robertson. My job is purely just to keep things on even keel until we have a hard stop at 4:40, 20 to 5. And we have 4 speakers from prestigious institutions: Cambridge, King's College, Edinburgh, and Oxford, and various other places actually they're affiliated to. Each one of them is going to talk for a short while, no more than about 10 minutes. And then we should have about 10 minutes for quick fire questions, and I'll try and compare those. So people who are thinking about questions, my suggestion is think about what are interesting questions that relate to the future of what we might do with all of this fancy AI jiggery pokery. What might that do for us? What new science might that open? And the 4 speakers have different slants on this, I think, that I hope they'll elucidate. There's Zoe Kurtzy, who'll be talking about analytics over diverse neurodegenerative data. There's Dan Bean, talking about natural language processing and the Cockstack system. Kenny Bailey, talking about machine learning for genomics. And Aidan Doherty, talking about reproducible machine learning and wearables data. At least I think that's what they intend to talk about. And so I think between them, that covers an awful lot of the sort of things that, that we're interested in doing. And we haven't much time, so I won't stand on ceremony. I'll just introduce Zoe. Zoe, if you could do the first 10 minutes for us, please.
Okay, thank you very much, David. Just sharing the presentation. Okay, wonderful, great. Thank you very much for inviting me. I'd like to present today briefly the EDEN project. EDEN is a project initiated by Alzheimer's Research UK, and it stands for Early Detection of Neurodegenerative Diseases. So just to give you a little bit of background, when we look at the space of dementia, globally 50 million people currently are suffering from dementia. And only in the UK, the health and social care costs exceed 25 billion. So a huge socioeconomic and health economics problem. So the main actually struggles when it comes to diagnosing dementia is difficulties with precise and early diagnosis. Currently, we have about 20% misdiagnosis at early stages of disease, and there are many different subtypes of dementia that require different diagnostic and treatment pathways and have different survival expectation. So really, there's a huge need for transparent and interpretable and interoperable algorithms for early and precision diagnosis in dementia, for precise patient stratification that can have impact in drug discovery and the cost effectiveness of clinical trials, but also for the diagnostic and the treatment pathway for individual patients and their carers. And ideally, we want to be able to do this as early as possible, ideally before symptoms occur, and therefore we need low cost and non-invasive data. So AR UK launched EDEN as an initiative to address this challenge, and the aim of EDEN is to revolutionize the early detection of dementia-causing diseases. And the main goal is to develop a single integrated digital fingerprint tool for detection of diseases that cause dementia 10 to 15 years before symptoms occur. Currently, the diagnosis of dementia is done through rather invasive tests, for example, PET imaging and lumbar punctures. However, as we go earlier in the diagnosis, before any symptoms occur, we have to come up with tools that are tolerable, that are really user-friendly. So we are looking primarily at inexpensive and non-invasive technologies such as mobile and digital technologies. So Eden is aiming for to have a transformative effect through early diagnosis and detection in health checks and clinical trials. And we can do this by encouraging lifestyle changes, having impact on patient stratification, understanding disease processes better and with more precision, and developing treatments early. So these four aims are really the main key impact that Eden aims to have. To do this, we have brought together a diverse team, bringing experts across universities and across sectors, including technology providers, people involved in clinical studies, data scientists, and people involved in data infrastructure. And the program has a strong research arm, but also a tool development, product development arm. And we have already recruited several patient cohorts that will be working with us from the early stages of testing the functionality and the usability of the tools, all the way to large-scale research cohorts and primary care cohorts before the tool is fully validated and implemented into health checks. Now, we have done the first actually selection of tools to be integrated into this diagnostic fingerprint tool. And we are looking at smartphone applications that measure both active and passive cognition. We're looking at smartwatches for measuring physical activity, sleep, and heart rate. And also we are looking at EEG systems that allow us to measure neural activity together with movement, breathing, sleep, and heart rate. And our plan for the technologies is to first test this initial toolkit and then keep adapting it to new technologies that we are reviewing. So very much an iterative process for defining the toolkit that will give us the necessary data for making these very early predictions for neurodegenerative disorders. We're also working on algorithms from a machine learning perspective that will integrate biomarker data, gold standard data, together with a non-invasive cognitive data, digital technology data that we will be collecting on our cohorts. And this is really key in integrating this data and building validated tools that as we go forward, potentially will not need to rely on heavily on biomarker collection, but we can actually make predictions early based on digital technology data. And what you see here is from the first algorithms we can build, what we are looking at is reclassifying individuals, not anymore on clinical syndromic labels, but based on their individual trajectories, on their individual disease progression. And we can reclassify patients, some stable patients that potentially maybe have other comorbidities that cause misdiagnosis, and they do not have dementia pathology, versus patients that show this disease progression and may move slowly or rapidly to develop different types of neurodegenerative disorders. And the most interesting thing is that we are already testing our algorithms with cohort data that are pre-symptomatic before any symptoms occur. So at the core of our approach is really an iterative way of working. We selecting the technologies that will give us the necessary data that is non-invasive and can be integrated and compared to biomarker data. And we are building the algorithms that will allow us to identify which are the key technologies to collect data from so that we can achieve this single integrated fingerprint tool. We have an amazing team of both from clinicians and scientists. We are organized in 3 hubs: the clinical hub that looks after the cohorts, the analytics hub that looks after the platforms and the algorithms, and the digital hub that looks more into the technologies, with great support from a superb coordination team from AR UK. So I'd just like to close this very brief presentation. I look forward to your feedback and questions. by saying that Eden really is a cutting-edge project at the space of neurodegenerative disorders, aiming to create a cross-disciplinary platform where experts among different domains come along to put data and algorithms together and achieve a fingerprint, an integrated fingerprint tool that can then be integrated into health checks and make early predictions before symptoms occur for neurodegenerative diseases based on non-invasive technologies. Thank you for listening, and thank you to everybody involved in Eden and to the funders supporting this work.
That's wonderful. Thank you, Zoe, and your timing is immaculate. And I won't, so not to disrupt things, I'll move quickly on. Dan, your turn.
Yes, hi. So my name's Dan. I work at King's, and this is some of the work we've been doing on COVID-19 analytics on top of our Cockstack platform. So this is a list of the projects where I had some, mainly small, involvement. All of these projects are using EHR data from King's College Hospital in London. And you can see there's a range of topics, questions, most of which are now published. But in addition to all using this King's data, all of these projects, I think underpinning them is the real-time analytics capability we have from Cockstack, the natural language processing capability that's plugged into that, which our system is called MedCat, the governance to be allowed to do these projects at all, and then collaboration, so between our data science group and clinicians in the hospital. And I think that without any one of these four, we wouldn't have been able to do any of these projects, never mind all of them. So thinking about what do we need for the future, I think each one of these four is going to have some kind of role. Looking into Cockstack, so we're within an NHS Trust network here. You can deploy Cockstack wherever you want, but most of the deployments I know about are on the Trust side. So in our case, that's running within King's College Hospital. In Cockstack, you take data from across the electronic health record, structured and unstructured data, which goes into an Elasticsearch index in Cockstack, and at that point, it's searchable. So clinical user or research user can search for patients with heart failure and no mention of whatever medication, get a list of results, even if you're searching tens of millions of text documents. On top of that, we have an NLP capability, in our case MedCat, but you can within Cockstack plug in whatever you want. And MedCat gives us the ability to search by specific concepts rather than specific just keywords. So we're mainly using SNOMED now. You could search for a SNOMED concept instead of a particular text representation. In addition to MedCat, where the initial training is unsupervised, but there's also supervised training on top using MedCat Trainer, where experts can read documents and mark individual annotations as correct or incorrect, and also we label meta annotations, which is the context of that concept. So is this mention of heart failure recent or historical? Is it negated or not? Is it hypothetical? All of that can be manually labeled and train a model, and then that's available for research as well. All of this technically is open source, and also we have an NLP model trained on Mimic that we can make open as well. And there's various publications. Per project, and specifically for our COVID data, this is our overall workflow, which I'm not going to talk through in huge detail, is mainly pretty standard stuff, but I think it's important that it was a lot of work to get to a point that you can do a fairly normal data science workflow within a hospital on top of raw routine data. The other important point here is this wedge on the left, which is manual work. So although we've got all of our, we've got our neural network doing all the NLP, but there's manual work in this still, which I'll come back to at the end. Primarily that was for COVID, things like symptom onset date, where that's a relative concept. So complaining of a sore throat for 10 days, that's something that a person read the notes and said, okay, well, 10 days ago was this date. Or some systems that are not yet in our Cockstack index. Then otherwise, it's your fairly typical workflow, data cleaning, and then we get comorbidities from MedCat, NLP, and also current medications that are recorded in text on admission. Other medications come from structured orders, and then bloods, vital signs, demographics, just standard cleaning. The important point is that these wedges are not to scale. So a huge amount of the data that's available in any hospital record is text, because it's designed for a person to later read and not for a machine to consume. So that's why it's so important that we have this MedCat NLP capability. Per project, we apply various different filters. Maybe you only want the first blood test, maybe you want blood tests every day. And we end up with something like 1,700 patients in published analysis now. I think our total cohort in the end was around 2,000. So this is the workflow that we're using for Cockstack for COVID work, but it's also basically the standard approach that we would take to answer sort of routine clinical questions now. News 2 is one of the COVID projects that I'll talk about in slightly more detail, although still only 2 slides. But News 2 is a very widely used routine risk score, which was being used in the COVID pandemic, but nobody really knew how well it worked specifically for COVID. So we tried to answer that question, evaluate the performance of News 2, and also see whether, since we're using routinely collected hospital data, is there anything else that's readily available that could improve the performance of News 2 for COVID? And this work was led by you and Cara Kings. So we trained a model on the KCH cohort and then validated that model in 7 additional sites, 5 of which were in the UK. We considered loads of 20 parameters, several comorbidities, various demographics, 8 of which were ultimately selected in a Lasso model. So the all features model in red is those selected 8 features in addition to News 2. And what we see in terms of predicting, will a COVID patient require intensive care treatment or even die within 14 days of admission, News 2 itself is okay at a push, not great. News 2 at age is another benchmark we use as a kind of nod to the fact that in reality, a doctor is not just using News 2, they're also looking at other features of the patient, an obvious one being age. Our additional model did improve performance across sites. However, we did also see evidence that this model is miscalibrated and specifically was tending to underestimate risk in some cases. Very briefly, from some of our other projects, we showed early on that there was not an increased risk of harm for ACE inhibitors. And we did look at the impact of ethnicity on COVID outcome, in this case with a case control study where our hospital cohort was matched to local controls from primary care data. And we also looked at the previous year's emergency respiratory admissions for non-COVID across, I think, 5 different London hospitals. We found that Black and mixed ethnicity was associated with increased risk of admission, but not with worse outcome following admission. In contrast, Asian ethnicity was associated with increased mortality. However, we had very low numbers of patients, so the confidence interval on that estimate was wide. Finally, we did some work on biomarkers, looking at trajectories of various measures over time in COVID patients to see if there was any clear subtypes of patients which maybe would benefit from more targeted treatments. I think the advantages of this Cockstack MedCat setup basically are speed, flexibility, and scaling. We were able to do things very quickly, primarily because Cockstack was already up and running and is 15 minutes behind the live record in the hospital. So we could respond quickly during the pandemic. Part of that is because we were already up and running in the hospital. We already have long-standing collaborations, ethical approvals. So I think we went from running to sprinting essentially during the COVID outbreak. It's very flexible, so we can annotate any SNOMED. We have pre-annotated all the documents with the whole SNOMED ontology, and we can pull out whatever concepts, blood tests, vital signs, because we're working with the raw data. And we can scale. I mean, the scale of the COVID data was not a problem at all. Our latest non-COVID analysis in the hospital, we have about 1 million patients, several million documents. That's probably hundreds of millions, I guess, of blood tests in total. It's just not a problem technically to scale to that kind of amount of data. The problem is that alongside that scale comes more manual work, which I mentioned at the start. There was manual review of all significant dates and outcomes, which was a huge effort from our clinical collaborators. I have to specifically acknowledge James Teo did so much of this manual work alongside being, working in a hospital during a pandemic. There's also manual annotation to train the model. So on the right, we have an example of MedCat Trainer where it's highlighted concepts that it's recognized, and you can annotate correct, incorrect, historical, different experiencer. That's manual work. There's also validation of where we've detected, okay, we think this person is on an ACE inhibitor. We have to check a sample to make sure that that data is reliable enough. And there's also things like project management work and writing the papers. And again, thinking about for the future, it made me wonder if writing up these kind of results as papers is necessarily worth the overhead of putting a paper together, especially when you need to get something out quickly. So with that, my last slide is just acknowledgements. There's so many people involved in all of these projects. And as I said at the start, this was not possible without collaboration. The code for Cockstack is open, as is lots of our analysis. And I need to acknowledge we've used patients' data for all of this work. So that's everything from me. Thanks for listening.
Thanks very much, Dan. And without further ado, on to Kenny, your turn.
Thanks very much, Dave. I'm going to talk about the future of several projects that you've heard about already from my friends Callum Semple and Albert Tenesa and others during this meeting. So I'm an intensive care doctor based in Edinburgh, and I lead the genomic study and co-lead the ISRIC4C study. And I'm going to talk about how we can use the foundations of those studies, particularly the data, for future challenges, specifically detection of the next outbreak and finding therapeutic targets. So as you may have heard, if you joined that session from Albert earlier, we discovered 4 new human associations, human genetic associations with COVID. They're illustrated in this slide. I'm not going to talk about the details of that. Albert did that earlier, but the key thing is that the genetics is one of the few things that gives us a causal inference about the disease. So we can learn from what's known about these genes. For example, this gene OAS1 is a host antiviral gene. We can learn from that controlling the virus decreases disease, which was an open question at the time that we did this. It could have been that the inflammatory disease that just damages the patient's lungs was a consequence of some idiosyncratic immune response, like an allergic reaction. This gives us hope that antiviral therapy will work. And if you've really been paying attention to the recovery trial 2 weeks ago, for the first time, we showed that that was true. So recovery trial found the first evidence that antiviral agent, monoclonal antibody, reduces mortality. And of course, this finds several other specific therapeutic targets in the host immune response to tell us how to alter the function of that complex system in order to change the outcome. So what's the opportunity? The ISRIC4C study is another UK-wide study looking at hospitalized patients, gathering samples from potentially every single hospital in the UK. In practice, there are about 70 hospitals who contributed large numbers of samples and data from a much larger area. Right now, there are 211,000 patients, individual clinical data, carefully curated and entered contemporaneously by research staff in the hospitals for hospitalized COVID patients. Deep phenotyping data from another 2,000 patients. Right now, overlapping data from the COG UK study with viral genomic data telling us all about the pathogen for 22,000 patients, but very shortly we are going to have data from more than 500,000 patients, viral sequences, and the host genomes, which I've mentioned. And all of that sits in a single analysis platform on the Edinburgh Parallel Compute Centre, the EPCC, which houses the UK national supercomputer, so colossal compute capacity and curated data, which is of course governed by the contributors, but the main problem in getting data usable by people with necessary skills is persuading the contributors to share it without giving up all of their control and ownership. And we've done that with this system where we have two divisions, one between a trusted research environment, which is all the protections for non-consented patient data that you would expect, and this flexible compute space, which is pretty protected, but allows flexible use of software and really high performance computing. And this other division between the embargoed area, which is, for academic contributors to contribute all of their data immediately without fear that someone else is going to publish it before them, but everyone can analyze it, and the non-embargo data area for once it's published or once the data contributors are willing to release it. And that's been working very well. So the data resource is there and is potentially extremely powerful. Where I think this needs to go in the future is Genomic has already been sampling the most critically ill patients, the most sick patients in the hospital, actually since 2015, with relatively specific infections. We're extending Genomic now to include all patients who are critically ill with any infection. Sue Hill, in an earlier session, commented that we ought to be doing that NHS-wide, and certainly for the intensive care patients, we're going to be. That opens up the opportunity not only to discover therapeutic targets in the host genome, but also to discover new outbreaks of new variants of COVID, new infectious diseases, and antimicrobial resistance by integrating with pathogen diagnostics, by making sure that the advanced pathogen diagnostics that are available through COG UK and collaborating centres are applied to those patients. So if you're critically ill, you're really sick with an infection, we investigate that pathogen very carefully to find out what's new. This all requires consent. That's a established ethical principle and isn't going to change. And the resilience and capacity to do that in the NHS is through the research system. So research nurses are the foundation of all of this work. And it opens up the opportunity to do the two things I mentioned at the start: surveillance to detect new diseases and target discovery. And on surveillance, I'll just give you the observation that every outbreak since Nipah or SARS back around about the turn of the century, but including MERS and Ebola and COVID and swine flu, every single one of those outbreaks was spotted by an astute intensive care clinician spotting a cluster of similar patients in their unit. But if you imagine there are 6 different units in the area, cases presented in all of those cases to different units and were seen by clinicians who didn't see more than one case, and so they didn't recognize the pattern. We need to develop tools to enable us to detect those outbreaks potentially months earlier so that public health can squash them before they become global pandemics. And that, if that's successful, we won't need to do all of this work that we're talking about again. But there are still diseases that we do need to tackle that cause critical illness year in, year out. And here the power of genetics to find therapeutic targets has tremendous potential. And we've demonstrated that, as you'll have heard from Albert earlier, that there are actually 2 drugs now in the recovery trial that were inspired or supported by the genetic evidence that we provided from the genomic study. So they've gone straight across from the genetic discovery straight into large-scale clinical trials. That's unusual. We don't normally manage that. Usually we have a bit more work to do. And the work is probably machine learning. So identifying not just what the genes are or the mechanisms by which they alter your biology, but actually what the disease is. It will surprise many who aren't clinical to learn that critical care doctors, particularly of whom I'm one, don't really have diagnoses. We don't really understand the pathophysiology, the underlying biological molecular processes that are happening in the diseases that we treat. And so gathering data, and particularly potentially causal data like genetics, will enable us to do that. And then we'll be able to test treatments initially in phase 1 and 2 trials, and then large-scale trials like recovery. I'm going to stop by acknowledging some of the contributors to the project. There are actually 1,440 people named on this slide. As I mentioned before, they are primarily the research nurses who recruit patients into the study, on whom all of this depends. And with an apology to Dave, I'm going to take the opportunity to mention that we are recruiting. This data resource and the potential to use it is available to you if you go to the isaric4c.net website. Please come and join us. Thanks.
Excellent, Kenny, thanks. And now for the last but not least of our four, Aidan.
Many thanks, Dave, and thank you to everyone else for coming along to this session as well. And I believe there are huge opportunities in the future for us to transform our understanding of possible causes and consequences of disease and possible assessments of treatments for a range of disease outcomes through the use of wearable sensors. And in particular, then, these will also offer new challenges for the appropriate and trustworthy analysis of those via reproducible machine learning methods. And Zoe has presented nicely earlier around the I guess possible opportunities for wearables with respect to Alzheimer's disease. And indeed today, around 8 million people are walking about the UK with either an Apple Watch or a Fitbit or a Garmin on their wrists. And these devices collect a wide range of physiological traits, including activity, sleep, heart rate, and rhythms. And it really does represent a huge wealth of what, at least as of today, is largely unexploited information. So there are huge opportunities to work together with patients and members of the public to think about how we can, in future, integrate this very powerful health and wellness data into the range of other data sources that we have available across Health Data Research UK. And the use of wearables is becoming more embedded in some of our sort of flagship biomedical studies, such as the UK Biobank, and there are a number of analytical challenges associated with that. So in the UK Biobank study, each of the participants, we give a device like this that looks essentially like a research-grade Fitbit device. It measures how much people move forwards and backwards, side to side, up and down. It does so 100 times a second. So we get around 180 million data points per person per week times 100,000 participants in the current UK Biobank study. And there then is a challenge as to try and get some clinically meaningful measurements from these devices, and particularly trying to identify types of activity. So the intuition being that the signature of movement will be different for sitting versus walking versus driving. And it is here then that we are really excited about the possible uses of machine learning to help get at some more clinically useful phenotypes from these sensors. The intuition being that as a person goes about their day-to-day activities, we have our time series data from the device in the bottom, and we extract out a range of time and frequency domain features to try and capture the signature of movement of individuals, put them through machine learning frameworks such as, for example, random forests to make a prediction for, let's say, every 30 seconds if a person is walking, sitting, running, or in this case, rowing. And then we also use other forms of machine learning methods to take account of the time series nature of the data. So, for example, we use hidden Markov models then where, for example, intuition is, it's predicted a person is sleeping for 30 seconds, sleeping for another 30 seconds, bike riding, sleeping. We use the hidden Markov model then to smooth over the erroneous classification in the middle, whereby it's unlikely a person went for a 30-second bike ride in the middle of the night. How well do these methods currently work? Well, in the data validation data sets that are free living validation data sets, these methods performed around 90% accuracy or a kappa interrate reliability score of around 0.8 or thereabouts. So not perfect, getting pretty close to being highly to being perfect and are already highly usable. And these methods have been deployed to the UK Biobank activity monitoring data set, whereby we can then construct this 24-hour model of what human movement behaviours look like in free living environments, in this case, the 91,000 UK Biobank participants. Reassuringly, at 4 a.m, most of the population are asleep, and then during the day, there's, as expected, a large amount of sedentary behaviour. And the beauty then of integrating, and it's really important that these wearable data sets are integrated with all the other forms of data and modalities of data that are available across the UK health data spectrum. Here I'm showing an example, for example, the discovery of the first genetic variants associated with machine-learned, clinically ascertained phenotypes from wearable sensor data sets that opens up new avenues for discovery then around some of the pathophysiology then of movement and sleep related behaviours. And in addition then, by linking this up via the UK Biobank with health outcomes, it can help as well redefine our understanding of how physical activity is associated with cardiovascular disease. So we've always known that activity is good for the prevention of cardiovascular disease, but key questions have remained as to, well, exactly what levels of activity should I do? And is there detrimental effects to me doing higher and higher levels of activity? So using the accelerometer assessed physical activity or wearable assessed physical activity in the x-axis and their hazard ratio for incident cardiovascular disease in the y-axis, we can see, first of all, there's very strong dose-response association, so no hint of detrimental effects by being engaged in higher levels of physical activity. And secondly, in the past, we had assumed those who are most active would have around a 20% reduction in risk of developing cardiovascular disease. Using our more precise measurements from wearables, we're finding that the physical activity is probably twice as important as we previously thought for the prevention of cardiovascular disease. So these wearable devices open up huge opportunities across a range of diseases to identify risk factors and prevention strategies, and also to ascertain patient-relevant outcomes to evaluate which treatments are helpful and not. So lots of huge opportunities, but as well, there are challenges, particularly the use of machine learning methods across health data science. Because as a field, we lag behind other fields of machine learning, such as computer vision and natural language processing, and that we're not as good at sharing code as other fields. We're not as good in testing the robustness of our methods across multiple data sets. So there's a lot of work that we as a community need to do moving to the future to ensure that our analysis is reproducible and trustworthy. So at Health Data Research UK, we have a national project then looking at this question of how reproducible machine learning practices can be embedded in future UK health data science to support trustworthy clinical insights. And we're looking at a range of questions that are listed here in the slides, and to give a hint or a flavour of what we're doing. And the first question then, we're dealing with just a very fundamental issue of how researchers should report the use of machine learning in health data science. So this is work then led by Gary Collins at Oxford, whereby he and his team is developing the tripod AI guidelines. So tripod is currently used for risk prediction models, and this will be an extension of tripod then for the use of machine learning or artificial intelligence methods. We're currently well underway around the Delphi processes for this, and have got a lot of buy-in from major journals as well to ensure that we can begin to shift some practices in terms of ensuring that the use of machine learning methods, particularly for risk prediction in this case, will be appropriately reported. So there's a lot more work to do in this area. I'd be delighted to discuss it with anyone, and for others who would interest in these areas. And for wearables, we really need to integrate these into larger studies that are integrated with the other modalities. Of course, we need to do reproducible machine learning and to ensure trustworthy clinical insights. And there are wonderful opportunities for partnerships with data custodians, clinicians, and also in industry as well, to combine these wearable data sets with other healthcare modalities. Thank you very much for your attention.
That's great, Aidan. Thanks very much. And now, if we can come back to the main screen. I've got some questions which I will field to you as best as I can. I think given the time we may not get through them all, but I'll do my best to pick off a few. And so the name of the game is very quick answers, I suppose, just to let you know. So and some of these are pretty broad, so it'll be hard to be quick, I suppose, but do your best. So first one is for all of these things, if you had to think of a key advance in analytics in your area in the next, say, 5 years, what's the main thing that the techies doing analytics could do for you? Anybody got a thought? I'll just make this catch-all if you don't mind, because it would be too slow to go through absolutely everybody. Any particular things that you would imagine they don't share? Kenny, then I'll do it.
It's a fantastic question. I think integrating the different sources of data that we have about people, there's much that we know that links different classes of data, genetics, the results of blood tests, the clinical information you extract from using NLP from a clinical record. We know from domain-specific knowledge how some of those things interact, but encoding that knowledge in a way that enables you to read across different data types and really integrate them to unleash the power of them, I think isn't certainly not much done, and is a primarily a technical problem. Solving that, I think, would be transformative in medicine.
Right, Aidan, and then I'll do Zoe.
So very briefly, I think that the major advance in the next 5 years will be just having access to greater and more interconnected data sets that have ever more diverse sets of data available for researchers, because quite often how one gets greatest gains on using these machine learning methods is by putting in good quality data to them. So I think this is where the efforts of others across Health Data Research UK is really incredibly important, and to help facilitate the analysis that many of us in the analytics community then can plug in on top of that.
Zoe?
Yeah, I would say it's really critical that we can build gold standards for data, like Aidan was talking about, the wearable data. There's a lot of information there, but actually we don't know how it compares to biomarkers. Being able to, through machine learning, learn about these gold standards in terms of the direct comparison, but then the mobile data, the wearable data, can actually stand alone for precise disease prediction. And we need really to move also at the same time at the space of individualized and precision medicine. So our algorithms need to be looking at these individual trajectories rather than the kind of the clinical labels.
Good. Okay, so another question is, which anybody could pick up, is anyone looking at activity post-op? Many patients wary of getting up and going, but we've seen fracture neck of female patients, early mobilization improves outcome, that kind of thing. Anybody looking at that bit of it? I don't know if you are. Maybe that none of you are.
Very briefly, I've got a PhD student is looking at exactly post-op in knee surgery, but the studies we have at the minute are very, very small, and just a couple of hundred individuals. So if anyone else is interested in that, please do reach out, because I think we should collaborate as communities. We really ought to have these studies in many thousands of individuals, and to look at some long-term trajectories of recovery after surgery, and also to look at people that are fit for surgery too.
Okay, and next question I've chosen is to do with integration. To integrate the different sources, do we need a new AI learning approach? such as machine self-learning, so pushing on the integrative bit. Any thoughts on that? You may not have, in which case we just move on. Aidan again?
No specific thoughts, but I think having just good linkage is what we most need then to integrate these resources.
Okay, so another one is to do with sustainability. Maybe this might be a good one for Dan, actually. So how do you get this stuff to sustain when you've got these big systems with lots of integrated components? Who maintains them once they're developed? How do you get over that problem?
Yeah, I think, and it's a really, really good question, and definitely a big problem now that there's several people who have systems that are ready for use, but how do you take something that was developed in research to something that you could actually genuinely say to someone running a hospital, I think this would be a good idea for you to use day to day, when research is not set up to provide long-term software support, and you need to be able to have someone on the end of a phone that you can call if your server goes down or you've got a clinical problem. We have been able to get some funding, and I think there's more and more awareness that we need to actually have these kind of roles to support and make things actually usable long term. And just making your code open is great, but it's not enough. That doesn't make something sustainable and usable, and we need to start supporting that, exactly that kind of sustainability plan.
Okay, and another one, this one was directed at Aidan, actually. I think it might be relevant to everybody. So the question is, can academia, the question as stated is, can academia keep up with commercial interests of big companies? But I guess you might have different interactions with companies. What's the company role in this? And is that something that's useful, sustainable? You were actually asked, Aidan, so what's your view?
I'm happy to shoot first. So my feeling is that I think there's huge opportunities to work together. So in studies such as the UK Biobank, we like a uniform measurement. So then sometimes it's maybe not so helpful to work with Apple or Fitbit or Garmin or whoever. But then industry, we can never stop industry innovating, and they will be developing new sensors, new phenotypes. I think it would be remiss of us to turn a blind eye to all of that. I think we really need to work together with industry then and what we bring in to play then is this, that we've been able to appropriately analyze the data, work together with patients and the public, and to then appropriately associate it with, as Zoe mentioned, and with various other biomarkers, and of course the important thing is ultimately with clinical outcomes. So I think huge opportunities to work together.
Good, Kenny.
Yeah, I mean, I've had very positive interactions with industry. There are two things I think the academic sector does extremely well that we should celebrate, and we've really learned about from the outbreak. One is people trust their doctors and scientists who are working to understand their diseases. And when you're asking people when they're critically ill to share their whole genome sequence, for example, for research, sometimes we get a bit of resistance from the public, if we say that we're going to share the data with industry. Now we do, and most times we talk people around in order to accelerate discovery for the purpose of finding new treatments. But I think the trust that the public have in the academic sector is important. The other, probably much more important component is we learned around about the end of March 2020 that there is a colossal reservoir of academic capacity, which is really what's delivered most of what we know about COVID. You know, the disease itself and how to treat it has all come from academic research, much of it in the UK. And I think that probably is going to still be the case for the rest of my career, and I fully expect to be involved in several further pandemics during that time. I think we need to maintain the reservoir of capacity in the academic sector and the ability to turn on a sixpence and divert resources to where they're needed.
Great, thanks, Kenny. And time is not on our side. The clock has hit 4:41. So and we do have a hard deadline for this one. Although I would have liked to have talked longer, but I think I must thank Zoe and Dan and Kenny and Aidan for the talks. It's been great. They fit it together despite the fact we had not rehearsed, so that was very good. Thank you all, and thanks everybody for joining and for asking such very helpful questions. Thank you.